Alright, so this brief lecture is going to introduce Chapter 12, which is looking at the DNA molecule and specifically how DNA is replicated. Okay, the following here are the objectives for this lecture. You're going to see that it focuses on vocabulary associated with the DNA molecule and then how the structures of DNA are going to tell us the function of DNA replication, which is very important. Okay, so this sort of became a bit of an arms race in a way across the world. Everyone wanted to find out what this genetic information was. What is a gene? How do they work? How do they transfer traits from one organism to another, parents to offspring? And then how do those traits show up as the phenotypes that we see? For example, how does the gene for eye color produce blue eyes? So of all of these questions, it all centered around the molecule of DNA. And because of that, we need to look at the history of how DNA was discovered. It really focused on two main groups of experimenters. We're looking at Rosalind Franklin and then James Watson and Francis Crick. Okay. James Watson and Francis Crick in 1953, they were given the Nobel Prize for their information and for their discovery of the double helix structure, but they could not have done it without Rosalind Franklin. She was the one who made the X-ray image of the DNA molecule, and that's what helped them to put this whole thing together about exactly what the shape of the DNA molecule was. There was a lot of controversy around this because Watson and Crick were the ones who were awarded that Nobel Prize, and that came with a lot of fame and fortune, whereas Franklin, she didn't even get an honorable mention. So, if you are asked this question on a test, it is Watson and Crick who are given the credit for discovering the structure of DNA as a double helix. So what exactly does that mean? Well, here's a diagram from your textbook. Okay, you'll notice that there are some key pieces to the DNA molecule down here at the bottom. Oops, let me get my pen. Oops, I'm sorry. There we go. Okay, down here at the bottom, we have adenine, thymine, cytosine, and guanine. These are what we call the nitrogen bases. Okay, and we're going to introduce those in a second. So this T corresponds to this T, A, to this A, and so forth. Okay, when we group a sugar, a phosphate, and a base together, that's going to form a nucleotide. And remember, this was the monomer. This was the monomer of our nucleic acids. Let me write that again on there for you. There you go. So that is a monomer of nucleic acids. So all nucleic acids have to have this basic building block piece, and then they're going to be pieced together. So let's review them one by one. Okay, so these nucleotides, as I said before, we've got the phosphate, the sugar, and the nitrogen base. In DNA, the sugar's name is deoxyribose. Notice the OSE ending there. That is meant to tell you that it is a sugar. Okay, you do need to know the specific name of this sugar. If you would write sugar on a quiz or a test, it would not be deemed specific enough. The different nitrogen bases, A is for adenine, G is for guanine, C is for cytosine, and T is for thymine. Okay, these are the four nitrogen bases that are part of each nucleotide. So that means there are four different nucleotides that are found in DNA. Okay, we haven't learned about this yet, but there is a fifth nitrogen base. It's called uracil, but that is not found in DNA. That instead is found in RNA. And when we start talking about the structure of RNA, we'll get to it then. So DNA stands for deoxyribonucleic acid. Okay, it is a double-stranded molecule. So we have one vertical strand here, and we have one vertical strand here. A lot of the times we compare the structure of DNA to a ladder. So you'll see we have these vertical pieces, okay, and then we have the steps or the rungs of the ladder. So along the sides we have a sugar and phosphate backbone. So here's our sugar, and then you'll see our little phosphate molecules in between that. So it is both of those that make up the backbone. And then in the center, we're going to see the nitrogen bases as they pair together. Okay, they make up what we call the steps of the ladder. And there are two different shapes to our nitrogen bases. Some of them have two parts. Okay, you can see there's like sort of an extra pentagon hanging off of them, and some of them are just a single hexagon shape. Again, this has to do with their organic structure. If they have two rings, okay, one ring here and one ring here, we call that a purine. So A and G are both examples of purines. If they only have one ring or a single ring structure, then we call them pyrimidines. Okay, so one ring versus two rings. Okay, so now these are going to pair together following certain rules, which we'll talk about in a second. But it also helps to understand that when we put one purine and one pyrimidine together, it's always going to equal the same width. So I'll go over that in a little bit more detail later. Okay. Chargaff had done some research a few years before Watson and Crick, and he took measurements of DNA bases that were found in all living things, and the numbers were just very, very patterned, and he found that as many A nucleotides as there were T nucleotides. And so when he published his paper, he said, oh, well, that must mean they are the same thing, they equal the same thing. And nobody really understood or cared about his paper. However, when Watson and Crick were putting together their ideas, his paper became very important. And now we know, because it's not that adenine and thymine are equal to one another, but rather that they are going to bond together. So we always know that adenine and thymine will bond together, and then guanine and cytosine will bond together. So it's important to note here, I'm going to make a little note. Okay, adenine was a purine, whereas thymine was a pyrimidine. Okay, and same thing with G, that was a purine, whereas cytosine was a pyrimidine. So if we add the ring structures up, 2 plus 1 equals 3 ring widths across, and here we have 2 plus 1 equaling 3 ring widths across. So it doesn't matter, that we're not pairing them, purine to purine. By having one purine paired with one pyrimidine, the width of the DNA molecule is going to stay uniform all the way down. So if we undid the spiral, the ladder would be an equal distance apart each time. Okay? If we were pairing A and G together, then it would be 4 ring widths apart. But with T and C, it would be 2 ring widths apart. And so then your DNA molecule would be all sort of a different shape. But we pair one purine and one pyrimidine together, and that is going to give us an equal ring width. Okay. So what holds these base pairs together? Between two nitrogen bases, we are going to use a hydrogen bond. Remember, from Chapter 2, hydrogen bonds are a very weak form of a bond. They can break easily and they can form easily. So right there is your first clue as to how DNA replication is going to happen. It's going to involve the splitting of the two strands, and those hydrogen bonds being so easily broken are going to facilitate that. Okay. So quick review of chromosome structure. In prokaryotic organisms, we have a single DNA molecule that is circular, just free floating in the cytoplasm. Remember, there's no nucleus in prokaryotes. Okay? There's about 5 million base pairs, give or take, and that equates to about 3,000 genes. So they still have DNA. They have all of the, C's, T's, A's, and G's. It's just going to be a lot smaller piece of genetic information. DNA that's in eukaryotes, much, much more, okay? We have approximately 3 billion base pairs, and that can range anywhere from 60,000 to 100,000 genes, although that's a high estimate, okay? So what's going to happen is we're going to take that very, very, very thin chromatin, and we're going to pack it very tightly by coiling it and condensing it into our chromosome structure. So it's going to become even more coiled than it already is. So let's focus on how DNA is copied. Okay, this is replication. If you cover up 1/2 of a DNA molecule, you would be able to determine what the other side was. Okay, because of the matching, because of the base pairing, right? So if we looked at this column and we said, oh, A, T, G, C, okay, and here, let me just write a sequence up here for you. Okay, let's say the sequence of this DNA was A, G, C, C, T, A, C, G. Okay, if I asked you to tell me, well, what's on the other side, you would very easily be able to do that because you'd say, oh, well, Mrs. Trifulio, I know A pairs with T, so this has to be a T. G pairs with C. C, that's a C. C pairs with G. C pairs with G. T pairs with A. A pairs with T. T, so on and so forth. Okay? Because of the base pairing rule, we now understand one side or one strand of the DNA molecule tells us how to build the other one. Okay? We call this semi-conservative replication. Okay? Oops. Yeah, let me get a pen. Okay. The semi part here meaning half, and to conserve something means to save. So in the process of replication, we're going to save half of the original DNA molecule in each part of the new two molecules that are formed. So what's going to happen is we're going to need to break apart the DNA molecule. So we're going to split it right down the center. And where that happens, it's known as a replication fork, because we're going to have growth occurring on both sides. We're going to be building our strands in opposite directions. So just the last thing here, the steps of replication. Okay, you will have enzymes. Let me get everything on there. Okay, first off, you have the enzyme called helicase. We know it's an enzyme because it's ASE. Helicase is going to come in and it's going to break those weak hydrogen bonds. So now your DNA molecule that was held together starts coming apart. Okay, so each single strand is now separate from one another. This is in the original DNA molecule. Okay, the next enzyme that's going to help out is DNA polymerase. So what it's going to do is it's going to find free nucleotides in the cytoplasm and it's going to bring them to each strand and it's going to start building a new complementary strand. So, let's say on this strand it says ATCG. DNA polymerase would bring in, TAGC. Okay, those are the complements, and it would base pair them up. The same thing is happening on this side of your DNA molecule as well. So, for example, let me just figure out how to do this here. Okay, just draw a little replication fork at this point. Okay, this part of my DNA, let's say it's still intact. Alright, and sitting right here, this is my enzyme helicase. Which direction is it moving? This way, right? Think about the zipper analogy that we used Okay? As you pull on a zipper, it's going to take the metal clips that are being held together and it's going to allow them to separate. So you have to move towards where they're still held together. So helicase is going to be moving up in this case. Alright, so let's get some letters on here. Let's say we've got an A, T, T, C. C is just not happy. There we go. And G. Let's keep it very simple. Okay, before we even separate, we know that the other side has to be the complement of this already. Okay, so the G and the C. There we go. So the black is going to represent our original DNA. Now polymerase comes in, and polymerase on this side is going to add a T here, an A here, another A G here, and a C here. While at the same time on this strand, a different polymerase molecule is doing the same thing. T, A, using, again, those base pairing rules. Okay? Now, just take a look at both of those strands, and, whether you mirror image them or not, note the fact that they hold the same exact pattern. Okay? So here you can see ATTCG. Well, look at this, ATTCG. So we produce two identical DNA molecules. In addition, here's the semi-conservative part again. Each molecule of DNA that's made contains two strands. One that is original and one that was newly synthesized. We look at the other DNA molecule. We have one that was original and one that is newly synthesized. Okay, so we're going to review these steps in class by actually building our DNA molecule and modeling with 3D pieces exactly how replication occurs. If you have any questions, please email me and let me know.